Okay, so that's the picture of Arup Chakrabarty. And what is the adaptive immune system? I'm hoping that I can make this bigger. All right. Does not work very nicely. Sorry. So it developed over half a billion years ago to keep vertebrates from being attacked by pathogens. And basically, what you have is that once once a cell gets infected by a virus, for example, the virus starts to make its own proteins, and these proteins are encapsulated and come out in the form of many more copies of the virus. And the adaptive immune system has two arms to combat these pathogens. One part are the antibodies. And as we saw yesterday, the antibodies are really evolved receptors of T cells that fit nicely to spikes or some other part of the virus and inactivate it, preventing it from infecting the cell. But the problem is that there are lots of host cells that are already infected and they could keep producing virus. So the other arm of the immune system are the T cells, which identify the cells that have been infected and destroy them. And the way that they do that is that the T cell has a receptor that tries to identify. Okay, so the T cell has these receptors that sniffs out that the host cells that are infected and undergoes various programs to either destroy the cell or become a memory T cell so that it recalls this infection and is ready to combat it in future. So the question of how the T cell recognizes which cell is infected, the answer is as follows, that each cell, of course, has this proteasome machinery that takes various proteins that are past their useful date and cuts them into small pieces. These are peptides that are of the order of, say, 8 to 15 amino acids long. And then there is a machinery that loads this, that is maybe garbage, this garbage onto a molecule that is called major histocompatibility complex, MHC, and displays it on the surface of the cell. So basically, the receptor of the cell, T cells, identifies these MHC molecules that are like bowls. They're displaying these peptides. And on the basis of the strength of the binding between the T cell receptor and the peptide that is presented, it makes the decision that this is piece of a virus and this cell has to be destroyed. So this receptor of the T cell that has to make this decision as to whether this host cell is infected or not has to have several important properties. First of all, it has to be able to recognize many things, potentially viruses that were recently evolved, such as COVID, and there is no particular code for the peptides of COVID that are going to be presented. And the way that it does that is that there are a huge number of these T cell receptors of the order of 10 to the 11 per person. And we certainly don't have 10 to the 11 distinct genes to produce 10 to the 11 different receptors. So the way that evolution has figured out how to do that is to use combinatorics. That is, there are these genes that come in a number of varieties called V, D, and J, and there is a mechanism by which they are shuffled. Some V is selected, some D is selected, some J is selected randomly to make a receptor of a particular T cell. And then there are additional deletions, insertions, etc., that ultimately give a potential library of the order of 10 to the 13 different receptors that can be generated through this mechanism. And actually, the proteins responsible for the enzymes responsible for this, the RAG genes, were discovered by David Baltimore, who passed away 6 days ago and was one of the giants of the field. So this is an interesting mechanism because you have at birth generated of the, well, at childhood generated of the order of 10 to the 11 versions of these T cell receptors. So there are all of them distinct T cells, and many of them go through life without doing anything. So you are willing to sort of sacrifice that much information, 10 to the 11 things, hoping that some of them would find what they will bind to. Now, since they are randomly generated, the question is, why wouldn't they also respond to pieces of self-proteins that are generated through this process and displayed on the surface? And if they do, then that's a problem because you start to attack your own cells and have autoimmunity. So that's something to combat. And then again, the T cells that you have are very specific to a particular part of a virus, and that's the mechanism that we have for vaccination. All right? Maybe take 1 minute for questions at this point, since understanding this mechanism is important. Okay. Yes.
Just to clarify, you're saying so many different receptors that actually each T cell is different from
each T cell is a different entity, yes. They could have their own children. In fact, when you have a memory T cell, then it creates its own children, which have that particular T cell receptors. But there are 10 to the 11 distinct T cell types. So, okay, and they are kind of randomly generated, hoping to bind something. So they potentially may bind to self, and the process by which that is partly prevented is called thymic selection. So basically, there is this organ called the thymus, and what happens is that the candidate T cells are generated in the bone marrow, but they are not immediately released to the blood or lymphatic stream. But before they do so, they go and get tested in this organ called the thymus. So what happens in the thymus is that it's a special tissue where all of the potential different proteins that distinct tissues in the cell generate are presented. There are cells that are presenting self-peptides. And these candidate T cells, they just go around and sniff the T cells that are presenting self-peptides, and if they react to them, they are eliminated. And this process is called the negative selection, and the idea is that you remove the randomly generated T cells. There's actually also something called positive selection, that they should at least bind to one of the presented self-peptides with some moderate amount of binding energy, so that they are not completely useless receptors that don't bind to anything. Okay, so some time ago, we modeled this process as follows. We assumed that the T cell receptor is a sequence, can be represented by a sequence of peptides, and that the MHC is presenting the peptide to be recognized, and the peptide to be recognized is also some combination of amino acids. So basically, we have a stream of amino acids that are labeling the peptide, and a stream of amino acids that are representing the complementary part on the T cell receptors. And we assume that the binding energy is really a sum total of pairwise binding energies assuming that the T cell receptor and the peptide are kind of in contact, and the number one amino acid of one is acting with interacting with number one of the other, and so forth. And all you need to do is to specify some matrix of the binding between pairs of amino acids. We use one of the available matrices, Miyazawa-Jernigan, but hopefully the results that I give are not specific to this model of binding, this model of particular interactions, etc. And this is actually an interesting problem of extreme values, because what you do, at least in our model, is that we randomly generate a candidate sequence for the T cell receptor, and then we present it with a library of self-peptides. So let's say we pick 10,000 sequences corresponding to, say, peptides of length 8, and we look at the interaction energy computed in this fashion between our candidate T cell receptor and the presented self-peptides. So each one of these self-peptides, each one of these blocks is supposed to be interacting with of the order of thousands of these numbers. We calculated the various binding energies. If any binding energies is too strong beyond some threshold for negative selection, that T cell is eliminated. If none of them are above some lower threshold of binding for positive selection, they're also eliminated. And it is an extreme value problem, because all I need to know is what is the extreme of all of these values, and the extreme should lie between these two limits. So that allows us to do some computations later on. But anyway, what are the results? So what is happening in this figure is that the various amino acids that can go into the system, there are of the order of 20 of them, are labeled according to which one of them is the strongest binding on the left, and which one of them is the weakest binding on the right. And according to some measure that we are using in our model. And we ask, what is the percentage of that amino acid having been accepted after going through this process compared to the percentage that was initially present in the ensemble, that corresponds to the ensemble of typical amino acids in human, for example. And what we find is that this ratio depends on how many peptides you see in the thymus. And just for computational purposes, let's say we vary this number from 1 to 10,000. We find that when you are selecting against few, then what happens is that there is a preponderance of strong amino acids, because you need to overcome this positive threshold. While if you are selecting against many of the order of 10,000, etc., then you find that there is a preponderance of weakly binding amino acids. Because strongly binding, if you have a few of them, then you can easily exceed the negative selection threshold. Okay? So suddenly this gives us an answer to how these T cell receptors can be simultaneously self-tolerant and specific. The reason for the specificity is that since you are becoming self-tolerant against a large number of self-peptides and you are in this regime, then you have a preponderance of weakly binding amino acids. And in order to get to some threshold for recognition, so that the T cell recognizes something, you have to have lots of weakly binding amino acids at the right positions, so that the combination of them together allows you to exceed some kind of a binding. And if there is some kind of a mutation and one of the presented amino acids is changing, then you lose one of the links that you need together in order to achieve the required binding energy. Now, this was tested in mice, and indeed, one can generate these mutant mice that present only few self-peptides in the thymus, and they find that the T cell receptors of those mice are not particularly specific, and they can bind to lots and lots of variants. And there is potentially a clinical consequence of that in the context of HIV, which is something that mutates very rapidly in the host. And as a result of that, it is hard to get the immune system to respond because there is constantly new variants generated through mutations, and the immune system cannot keep up. But there are some particular cohort of patients called elite controllers that can manage to bind to various variants of the virus as it mutates. And it happens that those particular cohort of patients also have some particular variant of the MHC gene that only presents fewer number of self-peptides. So there is this connection against between self-tolerance and specificity. And not surprisingly, that cohort is also more susceptible to autoimmune diseases. Maybe another minute for questions. Okay. Now, this is something that is called usually the central tolerance. And okay, not having my own computer, some of the images don't show up, that's fine. Now, the thing is that if you think about it, there is no way that this process of thymic selection can be completely perfect. Because what is it after all? There's a bunch of cells that are presenting self-peptides, and there's this candidate T cell that goes around and sniffs some of them, sees whether it is binding to self or not, and if it does, it gets eliminated. But I mean, after a while, it has to be released. And if you are presenting 6 million different possible genes, it is impossible that a particular T cell would have seen every single one of them. So inevitably, some of the T cells that come out of the thymic selection process have never seen some combination of self-peptides. And this is, again, something that is known, that we have within us T cells that are autoreactive. So the question is, how does autoimmunity not occur much more prevalently despite the presence of these autoreactive T cells? Okay, let's go through some nice statistical physics calculation. One can show that quite generically, if something has not reacted to M copies of something, and the next time it is presented with something taken from that distribution, the chances that it will react to that has gone down to 1 over M plus 1. This interesting calculation, which shows you that T cells that have gone through thymic selection, which say of the order of 10,000 self-peptides, if they encounter a new random peptide, the chance that they react to that is of the order of 10 to the minus 4. Okay, now, if they encounter a self-peptide, the chance that they react to that is not 0. It is just reduced from this 10 to the minus 4 because some fraction of the self-peptide they have already seen in the thymus and not reacted to that. Let's call that reaction M over P, M being the number that you have seen, and P is the totality of the self-proteome that would potentially be represented. And choosing some value of, say, seeing 70%, which is actually rather high, essentially reduces this probability from 10 to the minus 4 by 70% to 3 times 10 to the minus 5. Now, if we look at some typical tissue, like your hand gets infected, etc., and ask how many T cells are running around, they're of the order of 40,000 distinct T cells. So if the probability that each one of those T cells reacts to a peptide from some virus or pathogen is 10 to the minus 4 multiplied by 40, 4 times 10 to the 4, it's of the order of 4 will be activated. But still, if you ask how many of them are activated to self, it is of the order of 1.2. So we proposed in around 2013 that really the decision that we are looking at a self or foreign peptide cannot be done by a single cell. So what we propose that there is some kind of a quorum sensing mechanism going on. That is the immune system responds not because one T cell has recognized the pathogen, but some combination Q of them have recognized the pathogen, so that essentially requiring that Q things have responded changes the probabilities. And for the calculation that I showed you, if rather than one cell, that was 4 versus 1.2, I require 10 cells to make the decision in quorum. The difference between the two factors is enhanced to be something of the order of 10,000. Okay, so this has now been established that there is some kind of quorum sensing mechanism. And the mechanism for that is as follows, that T cells, once they recognize some peptide and they start to respond, they release some hormone called IL-2, and they also consume IL-2. And so by this concentration of IL-2, they can potentially reach some kind of a quorum. But there are also some other types of T cells called regulatory T cells that just consume IL-2 without producing anymore. So the combination of sort of positive feedback of generating more IL-2, some negative elements that eat IL-2 can cause a balance that allows some kind of a threshold to occur. And indeed, this is something that was recently seen in experiment where they essentially looked at small cells where they could put a certain number of reactive T cells, and they found that the overall system started to have some collective response if the number of T cells that were activated and were producing IL-2 exceeded a certain amount. So this quorum mechanism, in addition to the timing selection, is together some way of eliminating autoimmune diseases. Now, the final part of the story that I will tell is that there is another interesting thing that you may have heard of. That is, there is a correlation between onset of autoimmunity and being exposed to some infection. So it is, for example, known that um, there is an Epstein-Barr virus that is connected to occurrence of multiple sclerosis, which is an autoimmune disease. And what the graph here shows is the potential for occurrence of multiple sclerosis in two cohorts, one in blue without exposure or infection by Epstein-Barr, and the orange where there is infection by this other virus. And clearly there is, and this virus is a persistent infection. It kind of stays in your body for a long time, and it increases the chance of multiple sclerosis. And more recently, there are sort of indications of onset of diabetes in people that have the persistent version of COVID, long COVID. And so the question is, why is there a difference between persistent infections, where the virus stays in your body for a long time, and acute infection, where typically the immune response has cleared it, and there does not seem to be a connection to onset of autoimmune diseases. So one of the things that our collaborator found experimentally was that if there is a T cell that potentially has some degree, the T cell receptor, I mean, as I said, it's really a question of binding. So there could be T cell receptors that bind strongly to some foreign peptide coming from the infection, but maybe slightly weakly below that important threshold to self-peptides. So self-peptides typically would not really cause any problem with this particular type of T cell receptor. What one finds is that if there is a T cell that has already been exposed to a foreign virus and has some potential cross-reactivity to self-peptides, the previous exposure to the foreign virus makes it more sensitive to self-peptides. So it could be that previously it was not reacting to self, but having been already activated through this foreign peptide, it now can react to self. So essentially, the idea is that we, in our model, see what happens after a previous activation has occurred. We retest our model of ensemble of T cell receptors to self-peptides with a lower threshold and see that together you generate a quorum or not. And what we find, not surprisingly, is that as you keep going through this process, you will have more and more chances of these cross-reactive T cells activating against self-peptides and increasing the number that is required to reach quorum. So this connection has some experimental support, and in the model it shows up, and we have suggested this as a possibility to be tested. There is, when we were doing this, we found something that we kind of did not expect, which is that we actually looked at all potential self-peptides from human as well as some pathogens. And not every sequence of 8 amino acids or 10 amino acids is presented by MHC. There are certain programs that tell you whether or not this combination of 8 to 10 self-peptides are potential things to be presented. So we passed the human proteome peptides through that mechanism to see which one of them were being presented. And we found that surprisingly, among those that are presented, there are some that are composed of very reactive amino acids, which means that they can activate a huge number of T-cell receptors. And so this mechanism of quorum would never work for this because there were some of these that could simultaneously activate of the order of thousands of T cells. And so we are sort of, we have a potential proposal for how that is going on. And these regulatory T cells that I mentioned to you are important element of that proposal, but I will leave that for future. And I guess it's good time to summarize. So basically, we have this nice branch of the adaptive immune system, the T cell, that recognizes infected hosts through the presentation by the MHC molecule of pieces of peptide that could belong to self or foreign, and the T cell receptor has to distinguish them. And one way that it distinguishes, one way that the autoreactive T cell receptors are eliminated is through the process of thymic selection, where negative selection eliminates those that are binding strongly. But despite this thymic selection, there are bound to be some T cell receptors that are self-reactive that escape. And there appears to be this quorum mechanism where the decision to mount in, an immune response is not made by a single T cell, but a collection of T cells that communicate to each other through passage of this IL-2 and achieve some kind of a quorum. And there appears to be another way for this system to break down, which is when you have persistent infection. And persistent infection, the infection keeps presenting to the immune system more and more types of foreign peptides. And there are more chances for T cells to recognize these foreign peptides. And if there is some crosstalk and between compatibility to self and foreign, this persistent infection could potentially lead to autoimmunity at some point in time. And as I mentioned, Arup Chakrabarty has been very influential in getting me interested in problems of the immune system. Eric Hughesby is the person who did the experiments on mouse and other types of the other experiments that I mentioned. And there have been over the years a collection of very good postdocs and graduate students that have been working with me on this stuff. Thank you.
Any comments or questions? Right.
So I have a bit of trouble with the quorum sensing.
Yes.
Because if the, you said that, okay, well, suppose that each T cell has seen only 70% of the self peptides, right? But I mean, was the idea that the thymus actually has 100%?
That's a very good point. There's
Because if they all see the same 70%, then the quorum sensing doesn't work at all.
Okay, so one of the interesting things is that in each of our tissues, there is a particular set of proteins that are expressed. There is no reason for, say, the heart to express some proteins that are relevant to the kidney. Now, within this thymus, there is a particular system, there is another protein that comes and tells each cells to express as many things as they can. Okay, right. So the thymic cells that are their job is presentation. They present everything that they can. I don't know whether that was your question, but that's part.
Nearly.
Yes.
So as I say, if all the T cells had seen the same subset of self-peptides, the quorum sensing would not work.
Yes.
Yeah, it would, because you would get 1 to the
Right.
Q rather
right.
than
Yes.
0.7.
Yes.
Well, but then what about the elite controllers? What is the idea that there's a significant reduction in the fraction of your self-peptides that are in the
Yes.
hypothalamus then?
So essentially, there is this proteasome. So these cells of the thymus have generated all kinds of genes, and the proteins are being chopped up. All of the different proteins are being chopped up, and the MHC molecule scoops them up and presents them on the surface. So the surface shows a whole variety of self-peptides. Now, these MHC molecules have to somehow bind to the self-peptides in order to present, and there are a lot of variants of MHC. So my MHC molecules, yours, we don't match up with each other. There's, again, some selection that is going on. Now, there are some of these MHC molecules that bind to fewer versions of self-peptides. So the elite controllers, who are also more susceptible to self-peptides, their MHC molecules are of the variant that has presented fewer of these in the clinic.
So it's actually more of a curiosity. So are there cases in which there is in fact a misfunctioning of the thymus gland?
Actually, none of us by this point have a thymus glands. It is something that you have in childhood, and basically it shrinks, and so all our T cells are the ones that we have in childhood. Are there people who in childhood have some disease that prevents the thymus to work? I don't know. Presumably they are very immune sensitive, and they may not live long, but I really don't know of a specific disease of the thymus.
Could I ask a question about the last part? So, you're proposing a mechanism to explain the correlation between persistent infections and autoimmune disease. Does this understanding help with designing therapies or treatments for this condition?
Okay, so I mean, there's question is whether knowledge of this. Okay, so potentially, okay, so the idea is that these regulatory T cells suck up the IL-2 that is related to achieving quorum. Now, these regulatory T cells also start to do their job if their receptors recognize something. So potentially, if we can figure out what are these foreign peptides that are responsible for binding to T cells that are kind of complementary to self, and figure out what that peptide is that is binding to the, that is responsible for that, maybe we can design regulatory T cells that are activated by that. And if they are released, they can suck up the IL-2 and prevent quorum for that. I don't know whether that's possible or not, but that would be something that this mechanism suggests.
This is very interesting, and I think we can continue some of the discussion in the roundtable later. But let's just thank the speaker again and